Our next speaker is Dr. Chad Haynes. Dr. Haynes received his Bachelor of Science degree from Virginia Tech and the PhD from the Department of Biochemistry at the University of Kentucky. Since July of 2009, Dr. Haynes has served as a Science and Engineering Technology Advisor to the DOE Advanced Research Projects Agency Energy, or ARPA-E as it's also known as. Currently, he is the lead of that program for management of the electrofuels research portfolio and supports a research portfolio budget of approximately $85 million. He also serves as a subject matter expert and a management consultant to the U.S. Biomass Research and Development Board, USDA Agricultural Research Service, and the Air Force Property Agency. Today, Dr. Haynes is going to talk about that very agency, the Advanced Research Project Agency Energy, a new paradigm in transformational energy research. Let's welcome Dr. Haynes.
Thanks, Jim. So I'll look to you maybe for a time. Okay. Well, good morning, everyone. It's my pleasure to be here with you today. Thanks, Jim, for the introduction, and kudos to Phil and Matt for the work that they're doing. It's great to see that that work coming along. So it's my intent today to spend some time telling you a little bit about ARPA-E and what the agency has been doing at the intersection of energy and biology. And so I'm going to provide some examples of some of the research projects that are underway there. And then hopefully use that as a springboard, right, to have a discussion about where do we go next, right, with this whole endeavor. So for those of you who may not know much about the agency, ARPA-E is a relatively new agency within the Department of Energy. This slide depicts the evolution of the agency, if you will, from its first idea, which was first articulated as part of a report back in 2006 called Rising Above the Gathering Storm. Most of you are familiar with that report. It called for a DARPA-like agency for the pursuit of high-risk, high-return energy innovation in the country. Stephen Chu, Secretary Chu, the Department of Energy, was an author of that study, and when he came to Washington at the President's request, it was one of the major, I guess you could say, chess pieces for moving ARPA-E into play. The agency was actually first authorized under the Bush administration under something called the America Competes Act back in 2007, but there were no appropriations at that time. And it was really the Stimulus Act that provided the initial bolus of funding to get the agency underway. And that happened in 2009, as you guys may recall. So, since that time, ARPA-E has issued competitive cooperative agreements and also a few technology investment agreements with about 180 projects across the country spanning the entire energy landscape. I'll provide some examples of that in here a bit, to the tune of about $500 million. So the agency, I would be remiss if I don't have a slide, but I'd be remiss if I didn't mention some of the leadership of the agency. The agency's director is Dr. Arun Majumdar, formerly of Lawrence Berkeley National Lab in Cal Berkeley. And also Deputy Director Eric Toon, my direct boss, who's the Deputy Director for Technology at ARPA-E. He is also on an IPA to the federal government. His job, like many of you, you do 2 or 3 jobs. His other job is the Professor of Chemistry at Duke University. One of the features that I'd like to emphasize about ARPA-E projects is that there's a real emphasis and focus on the development of a deployable technology over a very rapid, short time period, okay? Typically, this is a very high-risk, high-return type of endeavor. And ARPA-E fits in this niche by providing funding for these type of research opportunities that are typically viewed as too risky by other government agencies or by the private sector. So that's important to keep in mind, I think. So the mission of the agency is broad but very clear. The authorization states that ARPA-E is to enhance the economic and energy security of the U.S. and to ensure the U.S. continues to grow a technological lead in developing and deploying advanced energy technologies. And so there's 3 specific mission requirements that are in the legislation. 1 is to reduce energy-related emissions. 2, to reduce energy imports, notably the import of petroleum, and to improve the energy efficiency across the entire energy spectrum. So the core of this is the development of advanced technologies to take care of this. So I'd like to present a sort of a graphical depiction of how ARPA-E thinks about their projects and the future of the future impacts of these projects. So this is a typical technology development curve where you can see that the cost of a particular piece of technology or unit of technology, say a smartphone, decreases over time as the industry scales, as demand increases. So this type of function is driven by numerous technological advances, hundreds, thousands of patents. And they're critical for driving down costs, clearly. But these advancements are often incremental in nature. And the performance of said technology may also be limited by very real constraints, right? Materials could be one, thermodynamics could be another. You can't fight physics, right? So the way ARPA-E is considering, when ARPA-E considers developing particular projects, they like to think about how do we explore new R&D space to find new learning curves, right? New technology learning curves that initially might come into the market at a higher cost, but could be understood and expected to eventually outperform current state of technology, right? So this could be the development of next generation advanced batteries that can beyond essentially lithium-ion batteries, for example, which have a energy density maximum. So a little bit of background on what some of the projects are underway. This was a snapshot of what came out of the first funding opportunity announcement that was announced by the President back in 2009. For this funding opportunity announcement, the call was for any and all innovative energy ideas. 3,700 concept papers came into the agency that were all reviewed by about 400 reviewers. If there were any reviewers who participated in that, thank you very much. There was a tremendous amount of goodwill by the scientific community, by the academic community, to help ARPA-E evaluate all these concepts. This was the result of that first call. So 37 projects, or 1% of those projects, were funded to the tune of about $150 million. As you can see, there were 5 biomass projects, and there were a few projects that, as well, that involved direct fuels such as algae-based approaches. And I'm going to talk a little bit about those today. So since that time, the agency has also developed some focus programs that fit into one of 3 particular bins. Stationary power, obviously a major area for innovation, everything from innovative controls to carbon capture and sequestration to the development of new alternatives for rare earths minerals. High end-use efficiency, another big opportunity space. There seems to be a lot of low-hanging fruit there, but on the other hand, there's a tremendous need to innovate new technology in that space. For example, the development of thermal storage batteries could be a major changer for that area. And then transportation, where I'll spend a little bit more time talking about 2 programs in particular: the electrofuels program, which I'm most closely involved with, and then the petro program. So let's talk a little bit about biofuels. We've heard, I think Matt's presentation was a great sort of segue into this. For lack of a better cliche, biofuels are a tough nut to crack, right? And at the end of the day, it's all about economics. So this is a chart that plots the relative total production costs for various forms of fossil energy, fossil liquid fuels versus renewable fuels. And you can see that everything from Middle East crude to oil that's coming out from enhanced CO2 recovery, enhanced oil recovery, is cheaper than to produce than sugarcane ethanol. And so this is a challenge, right? So how do you manage to compete with production costs that are essentially lower? And I'd be also a bit remiss if I didn't mention that this plot is about 2 to maybe 3 years old. And so we've seen some changes actually that have been very significant in this chart. So for example, on this chart, there's no tight oil. So tight oil that's coming out of the Bakken formation, for example, that's being pumped right now is not represented on this slide. I read last night that there was a new permit in the state of Utah that will, for the first time in some 3 decades, permit the commercial production of oil shale. And that's not represented on this slide. What's not represented on this slide is probably a shift in the cost of GTL, mainly due to the low cost of the feedstock right now. So $2 a million BTU, right? I mean, this is unbelievable. This is very low, and so how does that feedstock cost impact the cost of the final fuel product coming out of GTL? GTL is very expensive. I mean, regardless of the feedstock price, the project in Qatar is at least a $30 billion effort to produce about 300,000 barrels per day. There they get the natural gas for free. So they're going to be able to pay off that facility. So that's an interesting consideration that that you have to think about to make those profitable. We know that other people are looking into the development of GTL facilities even here in the States. Shell and both Sasol are looking at doing and running some feasibility studies in Louisiana right now. So this is the environment that we're working in. I'd also argue that, and I would suggest that corn grain ethanol is not as expensive as this chart implies. This again is 2 or 3 years old. So, improvements in efficiency of gallons per bushel and also the increase in ethanol titers up to 20%, I think have also been very helpful in decreasing the cost of corn grain ethanol, which is a great trajectory for corn grain ethanol to be on, and I think it's been a real success story. But sort of building off of that, crop-based approaches to biofuels is challenging, and you all know this in the room, right? So the energetic losses that you take from every step, from the first solar photon that comes onto an area or acre of land are significant. And if you track that through, the amount of energy that ultimately ends up stored in the chemical bonds of the plant is roughly on the order of, say, 5 to 6% of the energy from the incident solar radiation that struck that area of land. So, of course, the consequence of this is that you have to have very large acreages in order to essentially collect all this energy. Now, that doesn't make biomass-based approaches a bad idea, but it certainly should influence our thinking about how we go about developing technologies to address some of these problems. And so that, I think, is where ARPA-E has tried to come in and in many ways be complementary to a lot of the work that you guys are already doing and have done such a wonderful job on. So, what are some examples of ways to help think about these problems, and what are some of the solutions? So one of the projects that came out of the first funding opportunity was a project to look at the feasibility of using macroalgae or giant kelp, which is a marine organism, as a feedstock for the production of isobutanol. This was a joint project between DuPont and a small company out in Berkeley, California called Bioarchitecture Lab. And there are many attractive features to this type of technology, in the sense that the feedstock can be deployed in the open ocean, so there's no competition for land. There's significant nitrogen runoff from essentially all of our coasts, so nitrogen fertilization is pretty much taken care of. And the plant doesn't have any lignin. Okay, the harvest index is also 100%. You can take all of the biomass. There's no requirement to leave anything on the land, so to speak. So, okay, that's great, but there are obviously many challenges to this. One of the interesting challenges is the carbohydrate structure in these organisms is very different than the carbohydrate structure in a land-based lignocellulosic plant. So these guys are composed of polymers of alginate and mannitol. Okay? We don't have any organisms, at least biotechnology-relevant organisms, that can ferment alginate and mannitol to ethanol. That's where these guys came in. And so in this collaboration, DuPont and Bow developed both an ethanogen and a butanologen to take these sugars to these fuels. And this is the cover of Science from, I guess, a couple months ago, where Bow first published on their E. coli ethanogen, where they identified and moved in a series of genes responsible for alginate transport and metabolism into E. coli to support production of ethanol from macroalgae. The other half of this project, Bow, working independently of DuPont, is deploying a hectare-size open ocean farm off the coast of Maine as we speak to collect real-time empirical data to support techno-economic analysis to determine and compare the cost of sugars from macroalgae versus lignocellulosic sources or corn grain. So we should have that data later in the year. It's a first-of-kind study, and we're obviously very interested in that. Many of you may be familiar with the plant biotech company Agravita. So Agravita has traditionally been doing a lot of work on increasing stress tolerance in biomass crops and also improving yields. One of the projects that we found very attractive, that ARPA-E was very interested in, is this integration of biotechnology to improve pre-treatment costs. We know that pre-treatment is a major cost component of lignocellulosic material. And so Agravita's idea was to conditionally activate intene-modified cellulases. These are endoglucanases and xylenases that have inserted in them a protein sequence called an intene, which is found in bacteria, that can be conditionally spliced out with pH and temperature changes. And they've been successful in incorporating, firstly, to identify and develop these intene-modified enzymes, which can be conditionally activated. That was sort of the one hurdle, and then to actually get them into switchgrass and then demonstrate that there is efficacy in the plant. And they've been successful in doing so. Their project is actually coming to an end soon, and they're in the process of trying to extend the development of this technology by teaming up with a processor, right, in order to do some larger scale trials, in order to demonstrate how much cost savings we could see due to decreased enzyme loadings. Many of you in the room know the challenges that we face with respect to the addition of exogenous enzymes. And if you do the math, it's actually really interesting. It scales about 1-to-1. So what I mean by that is if you sit down and calculate the amount of enzymes that you need, say to support 1 billion gallons of cellulosic ethanol per year, you need about 1 billion gallons of fermentive capacity in order to generate those enzymes. And so, like I said, it's about 1-to-1, so 30 billion gallons a year, 30 billion gallons of fermentation capacity. Right now, we don't have that. And the notion that we might have that anytime soon, I think is unlikely. So this is a technology that attempts to address that problem. Ceres is another crop biotechnology company. They've been around for about 10 years. They made a lot of their money initially by essentially just doing sequencing of plant genomes and licensing that technology to a variety of companies and other people. Their technology was to develop switchgrass and sorghum with nitrogen use efficiency traits, and ARPA-E funded them to number one, incorporate those traits, demonstrate efficacy in the greenhouse, and then to deploy those traits to 4 different APHIS-permitted field locations across the country. In collaboration with Oak Ridge National Laboratory, they're doing what, at least as far as I know, to be the most exhaustive carbon and nitrogen accounting of the system in order to hopefully demonstrate that this type of technology works. For those of you who may have been closer to this type of field of study, there has been some I don't know, controversy may be too strong of a word as to whether or not these type of technologies actually work in the field. And so we're very excited. Hopefully we can get some data on that. And that will be wrapping up also in the next year. The agency's taken a very hard look at algae, right? So algae is a very promising type of technology, but the costs associated with production are really challenging, to say the least. Open pond systems are constrained by the amount of evaporative loss of water that you have and the increase in salt concentration, not to mention contamination. Closed photobioreactor systems are, number one, they have very, very low power density, and they're outrageously expensive. And I don't mean to be dramatic about that, but we've sponsored many models and the development of many models about that. Everything from the replacement of the materials to the amount of energy that's required to run these type of farms is really, really challenging. So one of the things that ARPA-E did to help try to address this is combine two technologies that came out of the first funding opportunity announcement. One was the development of a cyanobacteria that's capable of producing laurate at an efficiency of about 4%, with a innovative bioreactor company called Bioenergy, which was a spin out of the University of Minnesota. This company has developed thin film, essentially latex materials that can entrain organisms into a single sheet while actually maintaining the metabolic state of the organism. So we know that those two technologies sort of on their own are functional. So ARPA-E is trying to bring these two together to see if we can't get to a system that would be competitive, a new system design, if you will, that might be more competitive than what we traditionally think about, either open ponds or closed photobioreactors. The end of this project, which has already been demonstrated, is upgrading of fatty acids to jet fuel via catalytic decarboxylation. That's done by NC State, and that's been demonstrated, and a lot of other companies have that capacity as well. So moving along, essentially all the technologies that I just talked about are photosynthetic-based approaches, right? So those are on the left side of the screen. We know that there are direct approaches to also making biofuels. There are photocatalytic approaches. There are opportunities to essentially use solar electricity to power catalysis and the reduction of CO2 to methane and methanol. One of the challenges with both of those type of approaches, again, very interesting, they typically utilize rare earths, very expensive catalysts, and they suffer from, at the moment at least, an inability for building carbon-carbon bonds with high specificity. 5 minutes, great. So ARPA-E, in thinking about this, wanted to try to explore some opportunities to improve the energy efficiency of biofuels. And so sort of exploring the white space that was available and through a series of workshops and working with many folks in the field, we realized an opportunity to utilize biological catalysts that are obviously a lot better at making carbon-carbon bonds in high yield and specificity as the catalyst for electrochemical reactions. And so that spurred something called the electrofuels program. And I'll just spend a few minutes talking about that. The electrofuels program, at the core of it is the use of chemolithoautotrophs. Okay? So autotrophic organisms, these are autotrophic organisms that have the capability of deriving energy from a variety of reduced compounds. Okay? Reduced metals, direct current, hydrogen, ammonia, hydrogen sulfide, to name a few. And they're in many ways fascinating organisms. They've never really been looked at as a platform technology for the production of fuels. But that's exactly what this electrofuels program intended to do. There's a diversity of organisms that are in the portfolio. One of the challenges, I think, initially when this program was under review was just how genetically tractable a lot of these organisms were going to be. There was really a lack of molecular biology tools for working in a lot of these organisms, but to our satisfaction, I guess you could say, I think there's been a significant convergence over the past few years in a lot of different technologies that have enabled rapid work in a lot of these organisms that nobody had ever worked in before. So just a couple of examples of some of the projects underway. OPX Biotechnology, which is a group out in Boulder, Colorado. They already have a platform technology for the production of bioacrylate from sugars. That's in partnership with Dow Chemicals. This is their other project that they have underway, the use of hydrogen as a feedstock for the autotrophic production of fatty acids, which can then be decarboxylated to diesel fuel. This is 500 mils of diesel fuel that was produced by Rostonia in their, with their technology. That was shown by Director Majumdar at the summit. Another approach is the use of electrochemically reduced CO2 to formate as both a source of electrons and carbon for the production of fuels. This is being done by Jim Leal at UCLA, who's been a pioneer in the development of synthetic pathways for the production of butanol in microorganisms. And then very intriguingly, we've also, we know from work by folks in the microbial fuel cell community that organisms can obviously act as a bioanode, okay? So they can kick electrons out, if you will, to, through respiration. But it's interesting that some of these organisms, you can actually switch the current, and you can switch the switch the electrodes, and these organisms can act as a biocathode. So they can actually accept electrons indirectly. Very interesting type of biology, and a lot of it we don't understand in the sense that we don't really know all the mechanisms for how electrons get into these cells. But we're also utilizing this type of an organism for the production of, by taking direct current and to fuels as well. So we probably only have a couple more minutes, if any at all. But if this is sort of a cartoon representation of biofuel production, where you have traditional biomass feedstocks on one side and direct solar fuels approaches or electrochemical approaches on the right, electrofuels sort of fits somewhere in between maybe an algae-based approach and a direct solar fuels approach. And so Jonathan Birbaum, who couldn't be here with you today, did some thinking around opportunities to develop new types of feedstocks that might slot in somewhere in between traditional sort of lignocellulosic-based feedstocks and algae, which gives you something more like a fuel in its native state. And so he spearheaded a program called PETRO, which stands for Plants Engineered to Replace Oil. And there's really sort of three pillars to this program, right? So optimizing photosynthesis, a dramatic metabolic engineering in these organisms, and the rapid scaling and movement of traits that from one generation to the other. And he had set up some initial metrics and in order to set the program up. These are some of the programs that are underway. So we have groups that are working both in oilseeds, C4 grasses, trees, and in some other crops as well. This is one example from the University of Florida. Lullaby pine trees, they produce terpenes under native conditions and actually can be chemically induced to produce terpenes up to about 3%, so I understand. This group is attempting to genetically engineer and increase the resin canals in these trees in order to increase terpene production from 3% to something more like 20%. And so this is an opportunity to slot into an existing forestry infrastructure for pine, where you can actually provide terpenes, and at the end of the lifetime of the plot, you can provide wood for pulp and paper. Opportunities to engineer oilseed crops to more efficiently capture energy by optimizing chlorophyll throughout the plant, and then opportunities to divert carbon away from sugar production. Sweet sorghum, you can increase or accumulate sugars to about 10 to 15%, and the concept in this project would be to divert those sugars to hydrocarbons via diversion through fatty acid biosynthesis. So this is a project that's being done by Chromatin in partners with the folks that you see here. So that's a very, very fast overview of some of the things that ARPA-E has been doing, again, at this intersection of biology and energy. And I would just like to say before I end my little presentation here is that I think it would be great to talk about, where do we go next, right? There are many opportunities for biology and energy. And some of them may be sort of indirect. We heard one example earlier about potential wastewater treatment, right? So wastewater treatment coming out of not only biorefineries, but out of natural gas wells, right? So, downhole biology is an interesting area that we're looking into. Bio-GTL is an area that the agency has begun to kick around. And essentially improving the overall carbon efficiency of biofuels is an area that we're very interested in talking with you about. As you know, when you ferment corn sugars, you release carbon. Is there a way to be more efficient with carbon and essentially turn whole biomass, capture all those carbons into methane? Obviously, you need a source of reducing equivalents, but again, when you consider the requirements and what we're doing with our land, are there ways to be more energy and carbon efficient with some of these technologies? So that's just some food for thought. And so thanks for your attention. I'll be happy to answer questions if I can.